Now to look at some of the antifungal drugs. Remember that the cells of the fungus are eukaryotic, and that causes a problem because in going after eukaryotic cells, a lot of times the host cells are going to be not a lot of times, they are going to be eukaryotic, and they have some of the same characteristics, so it could also cause harm to the host cells. Most chemotherapeutic drugs are designed to act on bacteria, and they don't work on fungus. So the problem, as I said before, if it's toxic to fungal cells, it could harm human cells. There are 5 main groups of drugs that have been developed to specifically treat fungal infections, low and slow. Some bind to fungal membranes and they cause the loss of selective permeability. Many are broad spectrum, and sometimes they damage the chitin in the cell walls of the fungus. For antiparasitic chemotherapy, we have antimalarial drugs that destroy the Plasmodium parasite at various stages in its life cycle. This would be quinine and chloroquine. We have antiprotozoal drugs that will inactivate some of the metabolic enzymes, Giardia, Trichomonas, and the Entamoeba histolytica, the sulfa drugs, and the tetracyclines here. And then the antihelminthic drugs will totally immobilize and disintegrate and keep metabolism from happening in these parasitic worms. They're broad spectrum. They disable the worm and paralyze the muscles, and they destroy the scolex, or the head of the worm, where they attach to the host. Selective toxicity is almost impossible because anything that is viral, they're parasites and they're obligate intracellular. So what these drugs need to do is to block penetration into the host cell, or block replication or transcription or translation of the genetic material. And this would be acyclovir and the AZT, the HIV drug. And they also need to prevent maturation of virus particles. And this is also HIV drugs, the protease inhibitors. Now, the drugs for treating influenza, the original ones that were used, the antivirals, aren't used anymore because the viral strains developed almost complete resistance. Relenza and Tamiflu are a little bit more broader spectrum, and they block an enzyme in influenza A and influenza B. Drugs for treating HIV infections and there's a lot of drugs and combinations of drugs that manage HIV. All of them are going to target a specific part of the multiplication cycle of HIV. And there are some drugs that inhibit or block every step in the virus cycle. Interferons are produced by our fibroblasts and leukocytes, some of our white blood cells, and interferons are therapeutic because they reduce healing time and some of the complications that come with infections. They prevent or reduce the symptoms of the cold or the papillomavirus. They slow progress of certain cancers, leukemias, and lymphomas. And they're used for treatment of hepatitis C, genital warts, Kaposi's sarcoma that you see in AIDS patients. Now, moving to drug resistance. This is an adaptive response where the microorganisms start to tolerate the amount of drug that would ordinarily kill them or inhibit them in some way. And so they become more versatile in their population, and they're more adaptable. And this resistance can be intrinsic or acquired. And that's the main problem. Drug resistance is the main problem for antimicrobial therapy, chemotherapy. How does it develop? Well, you get it after spontaneous mutations in some of your critical chromosome genes, or either you acquire new genes or sets of genes that have transferred from another species. If it's through any intermicrobial transfer, you're going to get resistance factors that are encoded with the drug resistance. They're the plasmids. Also, the transposons are going to be duplicated and inserted from one plasmid to another plasmid, or maybe even form a plasmid to the chromosome. And you remember transposons are the jumping genes. Which of the following effects do antiviral drugs have? They kill the extracellular viruses.